Hey everyone, and welcome. Today, we're diving into this explainer on the molecular saboteurs inside your medicine cabinet. We're going to look under the hood of the complex pharmacology of everyday painkillers. We're talking about drugs that most of us take completely for granted, and we're going to see exactly how they pull off their medical magic.
It's kind of crazy when you really think about it, right? You twist your ankle, it swells up like a balloon, it throbs, and you just swallow a tiny generic pill with a glass of water. But how does that pill actually know where to go? There's no GPS chip in there. To really answer this, we have to shrink ourselves down way past the tissue, down to the molecular level, to uncover the hidden biochemical saboteurs at work.
Here's the roadmap for our explainer today. We're going to hit the anatomy of an inflammation, the COX enzyme pathway, mimicking the natural substrate, acetaminophen, the odd one out, aspirin's irreversible blockade, and finally, profens and advanced NSAIDs. Let's get right into it.
All right, Section 1. Anatomy of an inflammation. Let's set the stage with the macro experience of tissue damage before we get into the microscopic weeds.
So whenever your tissue takes a hit, whether it's physical or chemical, your body kicks off this wildly coordinated biological response. Boom. Step 1, chemical signals like histamine and prostaglandins are immediately released. Step 2, your capillaries dilate, which lets blood clotting elements rush right in. Third, chemotactic factors attract these specialized phagocytic cells. And finally, those phagocytes get to work acting like a cleanup crew, consuming pathogens and cell debris. It's a vital healing process for sure, but if it's left unchecked, it causes excessive pain, swelling, and tissue degradation.
Looking at the tissue level really brings this to life. Notice that very first stage right there. The exact second an injury occurs, cell membrane phospholipids are hydrolyzed, and those vital molecules, prostaglandins, or PGs for short, are unleashed into the wild. These are the direct, literal culprits behind the localized pain, inflammation, and fever you feel. Moving right along, Section 2, the COX enzyme pathway. Let's see how this builds from the tissue level down into our microscopic biological machinery. So who are the main characters here? Well, prostaglandins are these super short-lived lipophilic acids. We're talking a lifespan of maybe 1 to 5 minutes, but man, they pack a massive punch when it comes to causing pain. But where do they even come from? Enter the cyclooxygenase, or COX enzyme. Think of COX as a literal pain factory. It grabs a fatty acid that's just hanging out in your cell membranes, called arachidonic acid, and converts it directly into these pain-causing prostaglandins. So the COX enzyme is the exact factory churning out the pain signals. Section 3, mimicking the natural substrate. Now that we've found the factory, how does pharmacology actually step in to shut it down?
Put yourself in the shoes of a molecular detective for a second. Your whole mission is to plug up that COX enzyme so it can't grab the arachidonic acid, effectively shutting down the pain signals. How do you design a drug to pull that off?
Well, it's basically a microscopic lock and key situation. You see that yellow molecule? That is our natural substrate, arachidonic acid, snapping perfectly into an amino acid inside the enzyme called RG120. To stop the pain factory, scientists realized, hey, our drugs just need to impersonate arachidonic acid. They sneak into the active site, bind RG120, and bam, the natural process is completely blocked.
Which brings us to Section 4, acetaminophen, the odd one. Because as it turns out, not all everyday painkillers play by the exact same rules. Let's look at Tylenol. Acetaminophen is a para-aminophenyl derivative. And honestly, chemically speaking, it's actually kind of a weak general COX inhibitor. But the real kicker is that it's highly ineffective in environments with high oxidative stress or high ROS levels. What does that mean for you on a practical level? It means while acetaminophen works absolute wonders in the low-stress environment of your brain to fix a headache or drop a fever, it is effectively useless at reducing the localized swelling in a highly inflamed, stressed-out sprained ankle. It entirely lacks true anti-inflammatory properties. But this quirkiness leads us to a pretty dramatic clinical question regarding its safety profile. If someone takes way too much acetaminophen, hospital workers have to immediately administer sulfhydryl-containing compounds like N-acetylcysteine to save them. But why? What exactly is going wrong in the liver? Well, tracking its metabolism exposes a pretty dark side to this everyday drug. When you take a normal, safe dose, your body clears it, no sweat. But during an overdose, a minor yet critical liver pathway goes into absolute overdrive, converting the drug into a highly electrophilic, supertoxic metabolite known as NAPQ. This nasty little molecule will aggressively attack hepatic proteins, triggering severe liver failure.
Normally, your body uses a tripeptide called glutathione, which contains that lifesaver sulfhydryl group, to neutralize NAPQ. But in an overdose, you literally run out of glutathione. And that is exactly why doctors have to rush in with those sulfhydryl antidotes, to restock your liver's defenses before it's too late.
All right, let's switch gears. Section 5, aspirin's irreversible blockade. We are moving from Tylenol to a salicylic acid derivative with a totally wild mechanism of action.
Okay, so the biggest takeaway here is understanding how these drugs act over time. Your standard NSAIDs, they're competitive reversible inhibitors. They just kind of pop in and out of the enzyme's keyhole, competing with the natural acids. But aspirin performs a pseudo-irreversible covalent modification. To put it simply, while most drugs just visit the lock, aspirin essentially shoves itself in and violently breaks its key off inside.
If you look closely at this 3D complex, you can see exactly what I mean. When aspirin gets into the lobby area of the COX-1 active site, it doesn't just sit there. It actively donates its own acetyl group, that red structure right there, permanently modifying the serine 530 amino acid. This creates an inactive acetylated COX-1 enzyme. By permanently changing the architecture of the enzyme, aspirin completely and irreversibly blocks the natural substrate from ever getting inside. The enzyme is officially dead, and your body literally has to manufacture brand new COX enzymes from scratch to restore normal function.
Okay, final stretch. Section 6, profins and advanced NSAIDs. Let's wrap up our explainer by seeing how brilliant chemists purposefully tweak these structures to dodge nasty side effects.
Check out indomethacin on the right. It's an incredibly powerful anti-inflammatory, but it's notoriously harsh, causing severe gastrointestinal irritation. To solve this, chemists engineered the molecule on the left, sulindac. Sulindac is what we call a prodrug. You swallow it in an inactive form, and it basically smuggles itself past your stomach, relying on your liver to switch it on into the active drug later. By tweaking the structure and creating a bioisosteer, scientists were able to deliver the exact same powerful pain relief while brilliantly sidestepping the severe stomach irritation caused by indomethacin.
And actually, we really should break down exactly why these drugs hurt the stomach so much in the first place. Because NSAIDs degrade the stomach lining through two completely distinct pathways. The primary pathway is just straight-up local irritation from the highly acidic nature of the pills, resting in your GI tract. Prodrugs like sulindac dodge this by staying inactive when swallowed. But then there's the secondary, systemic pathway. See, your stomach desperately relies on the production of a specific prostaglandin, PGE1, to maintain its protective mucosal lining. By systemically shutting down the COX enzyme factory across your entire body to stop your sprained ankle from hurting, you are accidentally turning off the protective mechanisms in your stomach lining at the exact same time. And that can lead to severe ulcers and GI bleeding.
Which leaves us with one final, kind of scary thought. Remember, these NSAIDs are strongly lipophilic acids. The second they enter your bloodstream, over 90% of the drug binds like superglue to plasma proteins, primarily albumin. They basically just hitch a ride on the proteins floating in your blood. But because they hog so much space on these proteins, if you take them alongside other highly bound medications, the NSAIDs will physically shoulder check those other drugs right off the albumin. That floods your system with dangerously active levels of the secondary medication. It is a profound drug-drug interaction risk. Honestly, I hope this molecular deep dive leaves you with a totally newfound respect for pharmacology and causes you to reconsider the sheer biochemical chaos you're kicking off the very next time you casually pop a generic painkiller. Thanks for hanging out with me for this explainer.